Welcome back to the Deep Dive. If you are the kind of learner who loves a good scientific mystery, well, you are in exactly the right place today.
Absolutely. It is a great one.
Because we usually think of new species being discovered in the deepest, most inaccessible parts of the ocean.
Right. Like those massive, specialized ROVs plunging into oceanic trenches.
Eliminating bizarre alien creatures in the pitch black. That is the standard narrative, right?
It is. That's what we expect.
But today's sources reveal a discovery that completely flips that script. We are talking about a discovery right under our noses in Japan.
Which is fantastic. It serves as this really profound reminder of how much of the natural world is still waiting to be seen.
Yeah.
We have this pervasive tendency in field biology to assume that if an environment is familiar, if it's extensively surveyed, we have completely cataloged its inhabitants.
Right. We think we know everything in our own backyard.
Exactly. But nature constantly proves us wrong, usually in the most subtle, easily overlooked ways.
And that is our mission for today's deep dive. We are going to explore how a brand new marine animal was discovered hiding in plain sight in the waters off Nagasaki, Japan.
Just swimming right alongside everything else.
Yeah. And we're going to unpack the fascinating scientific process required to actually prove to the global taxonomic community that it is a new species.
The burden of proof is incredibly high.
It really is. To do this, we are drawing from some excellent primary sources today. We have coverage from Discover Magazine and the highly detailed taxonomic breakdown from a recent publication in the journal Zookeys.
Both are great resources for the granular details of this find.
So get comfortable. Okay, let's unpack this.
The setting itself is really the perfect place to start.
Right.
Because the context of the discovery is what makes it so entirely unexpected. We are talking about the Nagasaki region.
An area with a massive history of maritime activity and marine research. This was not a multi-million-dollar deep-sea expedition.
No, not at all.
It happened during a routine collection. Researchers were out in the coastal waters simply collecting adult specimens from a very common ornamental family of jellyfish.
A completely known quantity in those waters. Local aquariums collect them all the time for display.
So they are just pulling up nets of what they think are the usual suspects.
Right, and when you are doing that kind of routine fieldwork collecting a species, you have seen hundreds of times before your brain naturally goes into a sort of autopilot.
Oh, for sure. You see what you expect to see.
Exactly. It's a well-documented cognitive bias. You are primed to see the standard physical traits of that specific ornamental jellyfish. You expect that translucent uniform body that defines the local ecosystem.
But then, a visual clue completely stops them in their tracks.
Yeah, they notice something off.
Some of the individuals they were pulling out of the water had these distinct brown spots on their bodies.
Which shouldn't be there.
Right. For you listening, think about it like this. Imagine looking through a perfectly clear window pane in your house. You look through it every single day.
It's totally transparent.
Completely. But one day, you walk by and you notice a pattern of distinct dark specks scattered specifically across the glass, specks that absolutely should not be there.
What's fascinating here is how the scientific method kicks in immediately when faced with that kind of anomaly.
You don't just jump to new species, right?
Oh, never. When a researcher sees brown spots on a creature that is definitively supposed to be perfectly clear, the first assumption is rarely a new evolutionary lineage.
Because anomalies happen all the time in the wild.
Constantly. It could just be a genetic mutation within a single brood. It could be an isolated pathogenic response, or even just a developmental quirk from hyperlocal environmental stressors.
It's a weird mutant jellyfish.
Exactly. Statistically, a mutant jellyfish with abnormal pigmentation is way more likely than an undiscovered species swimming alongside a known one.
But this wasn't just a one-off mutant in Nagasaki.
No, it wasn't. The scientists started finding these exact same spotted variants in the waters off Yamaguchi as well.
And finding that consistent pattern across multiple distinct geographic regions completely shifts the investigation.
It changes everything. It was the crucial piece of evidence that effectively ruled out an isolated anomaly or a simple localized variation.
It raises a massive red flag.
A huge taxonomic suspicion.
Yep.
They had to confront the reality that they had stumbled upon a cryptic species, something entirely distinct that had just been blending in for decades.
Or centuries.
Exactly.
But suspecting you have a new species and proving it are two very different things.
Oh, 9 day.
To get that proof required an impressive collaborative effort. We're talking about scientists from Fukuyama University teaming up with the Tsuruoka City Kamo Aquarium and the Saikai National Park Kujukushima Aquarium.
The inclusion of those aquarium facilities is vital here.
Because you can't just take an iPhone photo of a spotted jellyfish and declare a new species.
I wish it were that easy, but no. Modern taxonomy demands rigorous, controlled studies, and aquariums of this caliber are highly advanced aquaculture laboratories.
So they transferred the live specimens from the ocean into these controlled facilities to study them.
They used a really smart two-pronged approach.
Right, the static and the live study.
Yes. First, they preserved a subset of the wild-caught specimens in formalin solutions.
Formalin is basically the gold standard for creating the physical type specimens for registries, right?
It is. Formaldehyde essentially cross-links the proteins in the tissue. It freezes the organism in time.
Which is important because a jellyfish is overwhelmingly just water.
Exactly. If you just try to preserve it normally, the cellular degradation happens almost immediately. The bell just collapses. The formalin allows them to perform incredibly detailed static analysis under a microscope without the animal changing shape.
But that static analysis only gives you a snapshot.
Right, it's just half the puzzle. So for the other half, they kept the remaining specimens completely alive to observe their development in captivity.
And maintaining a 247 jellyfish nursery is not easy.
It is an immense logistical challenge. You have to perfectly mimic the oceanic conditions of the Tsushima Current, the salinity, the temperature gradients.
They even use those specialized chrysal tanks, right?
Yes, chrysal tanks are amazing. They create a continuous circular water flow so the delicate jellyfish stay suspended. If they hit a wall or get stuck in a corner, it's fatal.
Wow. But by keeping them alive in those tanks, they documented the entire life cycle.
Meticulously, they track the progression from the tiny benthic polyps all the way through the strobilation process up to the fully formed, free-swimming adults.
Why is documenting that entire journey from polyp to adult so critical?
Because two distinct, closely related species might look nearly identical as polyps but diverge completely as adults, or vice versa. They could have totally different polyps but look identical as adults due to convergent evolution.
So by tracking the whole life cycle, they built a watertight biological case.
They proved its developmental biology was fundamentally distinct from the known ornamental species. The divergence was baked into its entire life history.
Okay, here's where it gets really interesting. Let's talk about the physical anatomy of this creature.
The scale is what makes the initial discovery so striking.
Yeah, we are not talking about a massive, imposing animal here. When you look at the measurements in the Zuki's paper, the bell of this jellyfish measures between just 0.4 and 0.7 inches.
Barely half an inch wide.
A tiny, translucent half-inch bell floating in the massive expanse of the coastal ocean.
And on that tiny bell, they documented a mouth with 4 lips and linear gonads.
Which are typical features for that specific subfamily, right?
Yes. It shares those foundational structural elements with its close taxonomic relatives. That baseline morphology is exactly why it blended in so perfectly. To the untrained eye, it just looks like the common local jellyfish.
But we have to return to the defining feature, the thing that sets it completely apart.
The brown spots.
Right, and they aren't just randomly scattered pigment anomalies.
No, the anatomical study confirmed the dark spots are located specifically and consistently on its reproductive organs and its stomach lining.
Think about that. Imagine being a taxonomist, staring through a microscope at a completely clear half-inch jellyfish and pinpointing tiny brown spots specifically on its gastrovascular system.
It requires an incredible level of eagle-eyed precision.
It really highlights the meticulous work by Fukuyama University and the partnering aquariums. But even with all that visual evidence, the life cycle, the spots on the gonad's visual observation isn't the final word, is it?
No, because of phenotypic plasticity.
Where an organism changes its physical appearance based on its environment.
Exactly. Plasticity can sometimes mimic speciation. So to eliminate any lingering doubt, you need undeniable proof. DNA. They extracted genetic material and conducted rigorous molecular barcoding. They looked at mitochondrial DNA markers.
The cytochrome sec oxidase subunit, I gene, specifically.
Right, which acts as a highly reliable genetic fingerprint for marine invertebrates. They took those markers and cross-referenced them against global genomic databases.
And the DNA definitively confirmed what the morphological data suggested. They had a previously unknown species.
But the DNA did something else too. It placed the new species precisely within the phylogenetic tree. It proved that it belongs to the Malagasy group.
Which is a huge deal because the Malagasy lineage is rarely found in Japan.
Finding it in the Nagasaki region is a massive biogeographical anomaly.
It totally is.
It introduces so many new hypotheses. Why is a Malagasy species thriving there? Is it an ancient endemic lineage that just stayed hidden for millennia?
Or is it a recent ecological shift?
Exactly. Maybe ocean warming is altering the Tsushima Current and pushing southern species north? Or, since Nagasaki is a heavily trafficked maritime hub, maybe ballast water transfer from international shipping introduced the polyp stage from an entirely different ocean basin.
The DNA didn't just solve the mystery, it opened up a dozen new ones.
Which is the best kind of science.
Truly. So with the DNA confirming the new species, the researchers finally earned the right to give it an official scientific name, and they picked a fantastic one.
They really did.
The official scientific name is Malagasy michelin.
It is a brilliant designation. It captures the unique morphology while using a really clever cultural analogy.
Michelin, as in the restaurant guide.
Exactly. The specific epithet Michelin alludes to the stars that this jellyfish accumulates as it ages. As it matures from the polyp stage into a free-swimming adult, those distinct dark spots develop and multiply.
So it's accumulating spots like a high-end restaurant accumulating Michelin stars.
A half-inch jellyfish earning its Michelin stars in the coastal waters of Japan. It is highly memorable.
I love that. But it also received a beautiful local name in Japan, Amanogawa Karish.
The Milky Way jellyfish.
Yes, which translates so perfectly.
It is incredibly evocative. You just visualize those dark spots scattered across the translucent bell, resembling a constellation of stars across the night sky.
It's a nice poetic contrast to the rigid Latin nomenclature.
But both names celebrate that initial visual anomaly, the unexpected spots on the glass that kicked off the whole investigation.
If we connect this to the bigger picture,
it signifies something profoundly important about modern biodiversity tracking. This tiny creature confirms that Japan's coastal marine ecosystems, areas that have been fished, studied, and interacted with for centuries, still harbor entirely uncatalogued species.
it is humbling.
Very. It reminds us how much of our own biosphere remains fundamentally unknown, even in places we consider completely mapped.
So what does this all mean? Let's quickly recap this journey. We started with a routine day collecting common jellyfish off the coast of Nagasaki. But because researchers broke through their cognitive bias, they noticed a subtle variation. A few localized brown spots on a half-inch organism.
Just a tiny detail.
That tiny detail sparked a massive collaboration. They preserved type specimens, they engineered complex chrysal tanks to culture them from polyps to adults, and they sequenced their mitochondrial DNA, all resulting in the Milky Way jellyfish, a species hiding in plain sight the entire time.
This raises an important question. What stands out to you? For everyone listening to this right now, consider your own daily routines, the environments you navigate, or the data sets you analyze at work.
How often do we overlook things?
Exactly. How often do we dismiss tiny data anomalies just because we assume we're looking at something familiar? We are neurologically wired to see the patterns we expect to see.
We stop critically evaluating our surroundings.
We do. But the true breakthroughs happen when we force ourselves to stop, break the autopilot, and question the unexpected spots on the glass.
Which leaves us with one final, lingering thought to ponder today. Think about the fact that this Milky Way jellyfish was discovered actively swimming among a highly common family of jellyfish.
Humans were already collecting them regularly.
They were in local nets, circulating in aquariums. They were an everyday occurrence. So how many other seemingly known and everyday items, plants, or animals residing in our own backyards or hiding in our existing data sets are actually completely undiscovered species?
They might just be waiting for someone to finally pause, look a little closer, and recognize their unique brown spots.
A perfect provocative thought to end on. Keep your eyes open and never assume the familiar is fully understood. Thanks for joining us for another deep dive. We'll catch you on the next one.